title of the talk is a little bit changed, and now is the neural oscillation and the synaptic plasticity. Let me see. Okay. First of all, I would like to very briefly to introduce my university. The name is Nankai University. This is the name of the district. You just mentioned the population. Now it's more than 10 million population in my city. Probably is something like in the Mexico City.
Because people think I am exaggerating all the time.
Okay, this university.
The other thing is that Nankai University is very well known to physicists. We are in physics department because it was the place where CERN was there.
Yeah.
In fact, the physics department is called the CERN Institute, the science and CERN's theories. Yeah. So it's a quite important place in physics. Yeah.
Thank you. This university was founded nearly 100 years ago, and however, during the Second World War, almost all the buildings destroyed by the Japanese. Only one building still there. This is a medical school. So this is a medical school. This is only one building after Second World War. And this is a gate of, this is a gate of our university. And this one is a memory for the associated university. During the Second World War, three top universities in China went to the inner district we called Kunming province. They combined, established a united university we called associated university. So this is just for memory. And this big bell is the memory of the Second World War. And now I very briefly introduce my college. The name is the College of Life Science. In this college, there are 5 departments. They are biochemistry and molecular biology, microbiology, genetics and cell biology, zoology, plant biology. And we have, this college was founded about in 1993, and we have about 170 faculty staff. Now we talk about before we talk about the neural oscillation and the synaptic plasticity, I have to say the human brain is one of the most challenging problems facing to the scientists and the medical communities. A big issue is how the brain works. And as we know, there are 2 fields. One is the brain science, sometimes we call neuroscience. Another is cognitive science. And the former usually focus on the material level, and the cognitive science usually focus on the functional level of the brain. The brain and the cognitive science ranged at several levels, which include molecular level, cellular level, and so on. It also includes several functions. For instance, perceptions, learning, memory, reasoning, language, understanding, and so on. So there are a lot of things we can do for the brain science. And in my lab, in this study, all of these components are involved in. Now, transfer to the neural oscillation and the synaptic plasticity. we got the three different stages. One is we interested the relationship between neural oscillation and the synaptic plasticity. And this is the first stage, and we got a lot of data, which some of which have been published. And then, if there is a relationship between them, then we are interested to know what is the underlying biological mechanism. So this is the second stage we're just doing. Now, the last one is just the hypothesis, whether or not neural oscillation synchronization based on synaptic plasticity. Okay, it's the first stage. We are examining whether the directionality of neural information flow could be used to index the measurement of synaptic plasticity. And then we are investigating which frequency oscillations are mostly related to the cognitive deficiencies. Then we are studying the coupling relationship of neural activities by measuring phase amplitude coherence between this specific frequency band, theta and gamma regions. This slide shows how the original idea of the study come from. If we say the cognitive functions such as learning and memory changed, the neurobiologist will be say, okay, such a change should be caused by the alteration of synaptic plasticity. On the other hand, the researchers for instance, neurodynamics will be say, okay, such a change should be induced by the alteration of neural oscillations. But we are interested to know what's the relationship between them. Now I would give a bit more words for the what is the neural synaptic plasticity. Simply say, neural synaptic plasticity is the ability of the connections. The connection is means two neurons connections. This connection strength will be changed in some conditions. And this figure shows there are two neurons. This one we called presynaptic cell. This one we called postsynaptic cell. And this is dendritic. This is the connections. If we gave some stimulation, for instance, high frequency stimulation, and then we collected the response on the postsynaptic cells, and we will find after the high frequency stimulation stopped, the response will be maintained at the high level. Such a phenomenon we called neural synaptic plasticity. And this is just how to quantitify, measure the synaptic plasticity. And this slide shows neural synaptic plasticity's molecular mechanism. I just simply introduce. If we give some stimulation, for instance, high frequency stimulation or another stimulation, then the presynaptic cell, there are lots of glutamines will be released. Then the sodium in the gap will be entered into the postsynaptic cell, which will cause depolarization, and then the magnesium will be ejected into the gap. A lot of calcium will be enter the postsynaptic cell. Then the cell will be more sensitive to glutamine. We call this process a positive feedback. So this is the molecular mechanism of the long-term potentiation, LTP. Now I would like to give a bit more words for introduce neural oscillations. What is neural oscillations? The neural oscillation is a rhythmic or repetitive neural activities which appear throughout the whole nerve systems. For instance, will be appeared in the cortex, appeared in the hippocampus, and so on. And also, physiologists divided the different frequency band that gave the name, for instance, for 4 Hertz to 8 Hertz, they defined the theta frequency band, and from the 30 Hertz to about 100 Hertz, they defined the gamma Hertz. And now the scientists understand, especially for the cognitive science, for learning and memory, for instance, the theta frequency band and the gamma frequency band are mostly involved in this kind of cognitive functions. This is our first stage. In the first stage, we try to understand or try to measure or what's the relationship between neural oscillation and synaptic plasticity. In the neural plasticity, we use an index. This is a quantitative measurement, which we call long-term potentiation, LTP. In the neural oscillation analysis, we use another index, which we call directionality of neural information flow, NIF. For doing this research, at first we have to set up our animal model. So in our lab, there are several animal models. I would like to introduce this one. This one is the depression animal model. How to set up such a depression animal model? I will give the introduction in the next slides. And also we have vascular dementia animal model, and also we have a melamine treated animal model. Now I would like to simply introduce how to set up animal model. We call this rat model is CUS animal model, means chronic unpredictable stress animal model. And the whole process of setup animal model use 3 weeks. Each week we give 7 stressors, which are food deprivation, water deprivation, electric food shock, reversed light-dark cycle, cake-tailed, ice-water swimming, tail pinch. This is all the stressors. Most importantly, we give the rats such a stress in random order, which lets the animal don't know which when kind of stress will be give it. Sorry.
Is there a rationale behind the kind of stress?
Mm-hmm.
What I mean, is there a specific zone in the brain that responds to pain or to electric shock or thirst? Or what is the rationale behind choosing these kinds of
my understanding,
stimuli?
yeah, my understanding is why in the modern society, lots of people depression because they don't know what's something will be hit him. So this is just simulated the human's depression. Right. So we gave the animal such a stimulation in random order.
But for instance, food deprivation is not something that is not expected. Either you eat or you don't eat. But a food deprivation probably puts you in the mood to be depressed.
Yeah. Yes. So this is the whole process. So 3 weeks, each week 7 stressors in random order. Gave the animal pain, basically. And this is our vascular dementia model. So I just show the slides. Sorry. Now, I would.
Sorry, I cannot. I want to understand that bit.
You may. Okay. First of all, we anesthetize the animal, and then we click the blood vessel here, then block, release several times to make the animal something happened. Cognitive function decreased or impaired. Yeah. So, and this animal model would take something like 4 weeks. Oh, really? So it's a long time. So, yeah. Okay. Now I would like to introduce the animal experimental method. This is all the experimenters we used to whatever set up animal model or doing some electrophysiological test. And experimental protocol was proposed and designed to answer the above 2 issues accordingly in vivo. So this animal is in life, not dead. So in life, what we call in vivo. Experiments were performed and then proper analyzing approach were applied. So I would now little bit introduce how to assess the cognitive function is damaged. So we use we call such a test is behavior test. And there are a lot of behavior tests. But in our study, we use called Morris water maze. Because rats can swim, but rats dislike swim. So we use these characteristics, rats characteristics, to perform our test. So this is all the just the situation when we do such a test. And we will collect several parameters. For instance, we will collect the escape latency. What this means, escape latency means it is a time and the animal take to find the platform, because the platform is under the water. The water is all black and all the circumstances is dark. The animal cannot find or recognize the platform based their vision. They have to use their spatial learning and memory. So this is an escape latency. Another 3 is 3% time percentage in target control and the numbers of platform crossing, swimming speed, and so on. So this is just to simply introduce the Morris water maze, by which we can measure the animal's cognitive abilities. And now I would like to introduce electrophysiological experiment. There are two electrodes in our vivo experiment. One electrode we called stimulation electrodes. Another is recording electrodes. Also, in our study, several neural pathways have been used. I just gave examples, and this pathway we called from the hippocampus to prefrontal cortex. This is a directional pathway. So in the neuroscience, some pathway is unidirectional, which means the information only from A to B, cannot back from B to A. But some pathway is directional. The information can both flow. And this one, hippocampus CA1 to prefrontal is bidirectional. So this is just examples, two electrodes, and this is the original trace of local field potentials by which we can analyze. Okay. Now I would very briefly introduce.
How many electrodes are in your silicon probe?
Sorry.
How many electrodes have you got?
Usually two.
Only two.
Only two. But one is we call simulation. But after stimulation, there still are recordings. So we recording the both area, the neural activities. This is the computational approach we used. This is the evolution map approach. This is a conditional mutual information. And this one is we call GPDC, General Partial Directed Coherence, which was developed based on the Granger consolidates. Now I would like a little bit more details for the algorithm of the EMA. The idea of the directional coupling is that if say system 2 is driven by system 1, then the evolution of phi 2, this is a function, the phi 2 will depend on the phi 1. In other words, prehistory of prediction of phi 2 from the previous value can be improved by taking into account the prehistory of phi 1. Only if system 1 drives system 2. In the formula, there are two parameters. One is C1, one is C2. So C1 and C2 is intensity of unidirectional, this is a one-way, unidirectional coupling respectively. And there will be normalized to the index D, which means directionally index. And this is the measurement of cross-frequency coupling. So it's a different frequencies. And in this study, we used modulation index, which we called PAC-MI, and also we used Z-score. Now I would like to introduce very briefly our preliminary results. They are divided into two parts. One part is identical frequency band. The second is cross-frequency resonance. So our first data were published in the neuroscience letter and the neural signal. In the paper in neuroscience letter, basically we used EMA approach to measure the direction of neural information flow over identical region between the cortex and the thalamus. An issue was addressed as to whether directionality of neural information flow could be used to index the measurement of synaptic plasticity in CUS, depression rats. The data suggests that the directionality index of neural information flow in identical frequency can be used to measure the synaptic plasticity.
Excuse me, I am a little bit lost.
Yes.
One, I think you make the measurement with 2 electrodes, so you have an average of many signals there, from say the thalamus to the cortex or something. And by these statistical methods, you could disentangle the directionality of the information flow. Oh, really? Very good.
Yes, yes. So this is we call the local field potentials.
Yeah.
Directly touch the neurons, and not just one neuron, so all the group of neurons, all the information we can collect it. So we call the local field potentials.
Yes, but those local field potentials are the result of averaging many individual signals and in many individual pathways of communication. So then you take the data and then you manipulate them to decide which direction is flowing and all that.
Yes, yes, yeah.
Good.
And now before we tested the depression model, now we talk about the vascular dimension. So the similar work in the vascular dimension model. But we changed the algorithm because in this study we find the two systems, the coupling strength is not the weak. Usually in the neural systems, the coupling between the two systems should be weak, but in this case, it's not the weak. So we have to change the algorithm. Now, so we selected the GPDC, by which the middle or a little bit high the coupling strength can be measured. This slide shows the decreased cerebral cortex connectivity by alteration of neural information flow in theta oscillation in depression model rats. This study examines which frequency field oscillations are mostly related to the cognitive deficiency in depression state. And our results shows that the effect of depression on LTP, long-term potentiation, impairment and cognitive deficits may be mediated by alterations in neural information flow in the thalamus and the cortex, which we call TC pathway. And this slide shows shows the alteration of face-to-face coupling. Now it's face-to-face coupling between the theta and the gamma rhythms in the depression state. And the data shows, we can see, the theta gamma phase locking between thalamus and the cortex is about, in the normal state, is about 1:6 ratio. However, it is almost diminished in the depression state, but will be recovered after the treatment. The melatonin, this is a drug we give the animals. So we investigated the coupling relationship of neural activities between thalamus and the cortex by means the phase interactions. Now the phase interactions between two algorithm oscillation in the depression. And this slide just shows the cross-frequency coupling of hippocampal local field potential was measured in vascular dementia rats, combined with the amplitude and the phase properties. We extract the signals both. One is the theta phase, one is the gamma amplitude. Then we measure their coupling strengths. So this is we call cross-frequency couplings. And we can see that in the normal state, there are quite strong cross-frequency coupling. However, it will nearly disappear in the in the vascular dementia rats. And this slide shows investigated whether or not directional index of neural information flow could be used to symbolize the synaptic plasticity impairments in hippocampal CA3 to CA1 network in rats of melamine. So this is a melamine, which we just changed the animal models. And we can see that in this is the normal state. This is the melamine state. We can see that in the melamine, in the normal state, there are quite strong coupling strands either in CA3 or CA3 to CA1 or the CA1. However, they almost disappeared in the melamine rats. Now we go to maybe a little bit. Now we go to the second stage. We interested to know what if there is a relationship between the neural oscillation and neural synaptic plasticity, and what is the underlying biological mechanism. In this study, we use the 4 animal group. One is a control, another is depression, and others 2 is the drug group, because it is reported that dopamine and 5-HT systems were mostly involved in the depression. So we use the dopamine receptor antagonist, just one group, and then we use 5-HT receptor agonist as another group. So this is the results I just very simply introduce. And we can see that the power in the low frequency band we call delta region in the CA1 was increased in the depression animal. In the, however, the power of the cortex in all the frequency bands were increased by the depression. And this is the synaptic plasticity measurement we call LTP. This is a time course. This is a control depression. This is a dopamine. This is a 5-HT. And in this one, we can see all 4 groups, the LTP were successfully induced. However, the other 3, depression, the dopamine, and the 5-HT, their LTP is significantly lower than that of the control group. And this slide shows the theta unidirectionally coupling strands. This is the control, this is a depression rats, this is a dopamine. The unidirectional coupling from CA1 to cortex was enhanced after external stimuli in this group. And this slide, we measured the cross-frequency coupling, CFC, between theta and gamma rhythms, and we used PCA modulation index. We can see that this picture shows the control group. In the control group, theta and whatever both lower gamma or high gamma, they are quite big coupling strands. However, in the depression, the lower gamma, theta and the lower gamma, no significant change, but theta and the high gamma almost disappeared. And these slides, we measured the effect of dopamine and 5-HT on phase amplitude coherence in CA1. These two, we can make the comparison, and we can see that the blockage of dopamine receptor didn't affect theta gamma coupling on either lower gamma or high gamma. However, the cross coupling between theta and the high gamma was significantly attenuated by the application of 5-HT receptor antagonist. And this slide, we also measure the effect of dopamine and 5-HT, but we used a different algorithm which we developed, because PAC modulation index, there are something we found is not very, there's limitations. So we developed a novel measurement which we called PAC PRV. PRV means a phase locking value measurement. The cross-coupling phenomenon measured by this normal algorithm was similar with that by the PAC modulation index. And in the control group, we can see that there is obviously phenomenon of the coupling strands. But in the depression state, nearly almost disappeared. So the average phase coupling strands of theta and the high gamma PAC was significantly attenuated in the depression group. So we can draw the preliminary conclusion. It suggested that the phase coupling was a crucial factor involved in the effect of CUS depression on theta gamma phase amplitude coherence in hippocampal CA1, and it might be achieved by the alteration of 5-HT systems and the expression of 5-HT receptors. This is just the funding. And I must go to my research students who have been involved in this study. Some graduates, some still in the lab. Okay, thank you for your attention.